Let's begin our journey with the fascinating discovery of ribosomes. In the 1950s, scientists using electron microscopy first noticed tiny particles in the cytoplasm and on the endoplasmic reticulum of cells. These particles, later named ribosomes by Richard B. Roberts in 1958, were soon recognized as the sites of protein synthesis. Over the next decade, researchers uncovered that ribosomes are made of two subunits and work closely with mRNA and tRNA to build proteins. Fast forward to the 2000s, advanced techniques like X-ray crystallography revealed the atomic structure of ribosomes, showing exactly how they decode genetic information and catalyze peptide bond formation. This groundbreaking work even earned a Nobel Prize in Chemistry in 2009, highlighting the ribosome's central role in biology. What exactly is a ribosome? Ribosomes are small non-membranous structures found in all living cells, both prokaryotic and eukaryotic. Their main job is to read the genetic instructions carried by messenger RNA or mRNA and link amino acids together in the correct order to form proteins. Think of ribosomes as molecular machines, composed mainly of ribosomal RNA and proteins, that translate the language of genes into functional protein. In eukaryotic cells, ribosomes can be free-floating in the cytosol or attached to the rough endoplasmic reticulum, while in prokaryotes they're found throughout the cytoplasm. Ribosomes are also present inside mitochondria and chloroplasts, highlighting their universal importance in life. Let's look closer at ribosome structure. Each ribosome consists of two subunits, a large and a small one. The small subunit helps position and decode the mRNA, while the large subunit catalyzes the formation of peptide bonds. Ribosomes are mainly made of ribosomal RNA, which provides both structural support and catalytic activity, along with ribosomal proteins. Are key differences between prokaryotic and eukaryotic ribosomes. Prokaryotic ribosomes are 70S, made of 30S and 50S subunits, while eukaryotic cytosolic ribosomes are 80S, made of 40S and 60S subunits. The S stands for Sveberg unit, a measure of how these particles behave during centrifugation. This distinction is crucial, especially when considering how antibiotics can target bacterial ribosomes without affecting human cells. Now let's explore how ribosomes actually build proteins, a process called translation. It starts with initiation, where the small ribosomal subunit binds to mRNA and the initiator tRNA. The large subunit then joins, forming a complete ribosome. During elongation, tRNAs bring amino acids to the ribosome, which links them together, moving along the mRNA strand. This continues until a stop codon is reached, signaling termination. Finished polypeptide is released and the ribosome is ready for another round. Ribosomes have three key sites. The site accepts new tRNAs, the P site holds the growing protein chain, and the E site is where empty tRNAs exit. This precise choreography ensures proteins are made accurately and efficiently. Are ribosomes so important? Simply put, proteins do most of the work in cells. They act as enzymes, receptors, structural components, transporters, and regulators. Without ribosomes, cells couldn't produce these essential molecules, and life as we know it wouldn't exist. Interestingly, many antibiotics work by targeting bacterial ribosomes, exploiting the differences between bacterial and human ribosomes to stop infection. So, ribosomes aren't just fascinating molecular machines, they're also central to medicine and biotechnology. Remember, ribosomes are the RNA protein machines that read mRNA and build the proteins that keep us alive.